PROTECTA Consortia was created with Horizon 2020 support, with the purpose of gathering several research groups focused in the study of oomycetes. As a well-articulated network, it will provide insights in the biology of these microorganisms and their hosts. The knowledge generated within the consortia will serve as a prime for the design of tools to manage global food security, biodiversity preservation, and to develop new sustainable disease control actions. Also, a new generation of scientists will be trained in PROTECTA Consortia thanks to the extensive training program designed for this purpose. In this multidisciplinary network, some of the most important European universities can be found. Following, we would like to introduce each of the participant research groups with a brief summary of the scientific projects. The Norwegian University of Life Sciences is located in a small community 30 kilometers south of the capital, Oslo. The Department of Plant Sciences at Faculty of Bioscience has the national responsibility for education and research on sustainable production. The Division of Biotechnology and Plant Health of the Norwegian Institute of Bioeconomic is part of the PROTECTA Consortium. This division has Norway's most comprehensive competence in plant health and plant protection in connection to both agriculture and forestry. The campus has a modern infrastructure, field and greenhouse testing facilities, advanced laboratory equipment for molecular biology, including transformation and protein analysis, as well as omics technologies. Bikal Ghimire is a PhD student within PROTECTA. His studies will focus on mining effectors in Phytophthora cactorum, the causal agent of crown rot disease in a strawberry. He is supervised by Professor May Ventet Brubergs. Professor May has special interest in the crown rot pathogen of a strawberry the last decade. Last year, she published the genome sequence of Phytophthora cactorum, including nearly 300 effector genes. Student Bikal will use various modern techniques like genomics, proteomics, gene expression, protein-protein interaction, genome editing, and many more.
Hello, I'm Bikal Ghimire and I come from a country with many beautiful mountains called Nepal. With an aim to be a plant scientist, I did my undergraduate study on agriculture science and my graduate study on crop protection and sustainable agriculture from University of Göttingen in Germany and University of Padova in Italy through Erasmus Mundus joint master's degree program. Having done my master thesis work with the effectors of fungal pathogen Alternaria solani inspired me to work more with effectors to understand disease-resistant mechanism for future exploitation in crops. And I feel very happy to have been enrolled in this project in which I am mining the effectors of oomycete pathogen Phytophthora cactorum that causes crown rot disease in strawberry, which is one of the very highly valued fruit crops. However, the loss in production due to crown rot is one of the severe problems in strawberry farming. Even with extensive use of chemicals, Phytophthora cactorum is very difficult and costly to control because of the survival of oospores in soil for many years. Furthermore, the use of chemicals is perceived negatively by consumers, industry, and it creates huge environmental burden. Identification of pathogen effectors can help us to explore plant-resistant proteins that trigger immunity in the plants. Hence, my strategy in this research is first to knock out candidate effectors, perform infection assays, and establish functional validation system for effectors and receptors. Some of the important tools and techniques that I'm going to use in this research project are agrobacterium-mediated transient expression in plants, for which I'm growing some tobacco plants, CRISPR-Cas9 based gene editing techniques, activity-based protein profiling, effector localization, quantitative proteomics, and bioinformatics. And I believe the findings of my research will benefit plant science community and strawberry farmers to have sustainable resistant strategies against Phytophthora cactorum and the society of consumers of which we all are a part of. I am very much thankful to European Commission for funding this research project. Thank you very much.
The next project is being developed in the Forestry Faculty of Swedish University of Agricultural Sciences, located in Alnarp in southwestern Sweden. The Forest Pathology Lab studies the biology, ecology, and epidemiology of endemic and exotic invasive forest pathogens. Here is where the PhD student Noelia is developing her research. Johanna Witzel, Noelia Supervisor Research, focuses on forest diseases, tree resistance, and stress responses, with a particular interest in phenolic defensive compounds, functional traits of fungal endophytes of trees, Dutch elm disease, and invasive Phytophthora species. The lab work focuses on aspects of disease control and the interactions of trees with fungi and oomycetes. Also at the Swedish University of Agricultural Science, Jennifer and Christian are PROTECTA students in the Integrated Plant Protection Unit. They study the biology and ecology of pests and their natural enemies in agricultural and horticultural crops, both in field and greenhouse conditions. Professor Laura Greenfield-Briggs-Didymus, the head of the unit, is their main supervisor.
I'm Jennifer Simati from India. I graduated from Wageningen University and Research in the Netherlands with a master's in plant biotechnology. I specialized in molecular plant breeding and pathology. With deeper understanding about the effectors in Phytophthora infestans, we can develop better potato cultivars. So, my project will mine effectors from various infestans isolates and functionally characterize some. We will also study the interaction between microbes by characterizing the infestans effectors when it is being attacked by the oomycete Pythium oligandrum. Through this, Pythium can be efficiently formulated to act as a biocontrol. As a whole, my project will help to integrate breeding and biocontrol to mitigate potato late blight. Cool techniques like Bensec enrichment sequencing, stem loop PCR, transient gene silencing, and confocal microscopy will be used to achieve these aims.
Go.
My name is Christian Benjamin Andersen. I graduated with a Master in Agronomy from Copenhagen University of Plant Science. My main focus was plant pathology, specifically focusing on biological control of plant pathogens. My main research is focusing on the oospore biology, effector characterizations and analysis, and the ecological risk assessment of the oomycete Pythium oligandrum when used as a biocontrol agent to manage several severe phytopathogens such as Phytophthora infestans and Aphanomyces cochleoides. My research will contribute to a more sustainable and integrated approach to control plant diseases. This, however, will require obstacles to be overcome, one of which is the efficacy of the biocontrol agent Pythium oligandrum when deployed in the field compared to conventional pesticides. Hence, this is why an understanding of the fundamental biology of the oospores is of crucial importance. In order to achieve all of these goals, a better understanding of this complexity, we will use a holistic scientific approach and utilize a series of novel pathology techniques which include transient transformations of oomycetes, confocal microscopy, next-generation sequencing, metabarcoding, and GCMS.
Hey, my name is Noelia Lopez Garcia. I graduated with a double master degree in Mediterranean Forestry and Natural Resource Management from the University of Lleida in northern Spain and the University of Lisbon in Portugal. I recently started my PhD project within the PROTECTA training program. It will focus on forest Phytophthora, ecology, diversity, and management. Within my project, I will map Phytophthora species composition in environmental samples collected from forest environments that represent a gradient of humans' influence. Then, I will quantify effector gene expression of selected Phytophthora species isolated from the same forest settings, and finally, I will provide data for pest risk assessment for forest Phytophthora at the European level to develop appropriate phytosanitary strategy at the local level. To achieve my main objective, I will use a holistic scientific approach involving techniques like painting, qPCR, PCR, biomark, fluiding, chips technologies, metabarcoding, and next-generation sequencing.
Also at the University of Aberdeen, the research activity at the School of Biological Sciences is organized through 4 key research clusters with an international reputation for cutting-edge research: ecology, environment, evolution, and ecophysiology. These programs affiliate staff and students with shared research interests, facilitate multidisciplinary ways of working, and incorporate seminar programs, symposia, and workshops for fostering collaboration and interaction. The Professor Stephen Woodward has extensive collaborations within EU projects and other networks. The main research carried out in Stephens Woodward's group is focused on invasive pathogens, their detection, the damage they cause, how they are spread globally, and mitigation methods that can be used to reduce their impacts on natural ecosystems. In his laboratory, a range of pathogen problems is under investigation, currently focusing on species of Fusarium, Verticillium, Phytophthora, and Pythium, and the potential use of bacterial biological control agents against diseases caused by fungi and oomycetes.
Hello, my name is Clara Benavencelma. I am from Spain and I studied forestry engineering and a master on plant protection in Valencia. My research experience previous to the PhD has been in the control and management of agricultural pests. Regarding my project, it has been recently studied and confirmed in our group that the international commerce is one of the main dispersal pathways for the introduction of plant pathogens, being reported in the last decades the introduction of new plant pathogenic Phytophthora species. So the aim of this project is the study of eco-friendly strategies for control and management of Phytophthora species internationally, nationally, and regionally. And we are specifically focusing on the study of Pythium oligandrum as a possible biological control of Phytophthora cinnamomi. For such a study, we are using both classical mycology methods and advanced molecular techniques, carrying out experiments both in vivo and in vitro. Thank you.
Maribor Hilleshock is a global supplier of sugar beet seed. It was established in the 1st of October 2017 and has more than 300 employees dedicated to sugar beet breeding and seed production in Denmark, Sweden, Italy, France, and the US. The R&D activities are based in Maribor Hilleshock Research AB in Sweden and focused on the development of new and improved sugar beet varieties. Most of the research activities are conducted in the pathology lab and seek to develop new disease-tolerant cultivars to manage sugar beet diseases, such as Cercospora leaf spot, Endophanomycetes, and Rhizoctonia root rot. Dr. Louis Holmqvist is a trait integration leader at Maribor Hilleshock with a background in plant pathology and a PhD thesis on the soil-borne pathogen Rhizoctonia solani and sugar beet responses. The company has state-of-the-art plant growth facilities for the evaluation of plant diseases and modern laboratories to conduct molecular and microscopy analysis to study host pathogen interactions.
Hello, my name is Valentina. I have a master's degree in agricultural and environmental biotechnologies at University of Perugia in Italy, and my research project focuses on the oomycete Aphanomyces cochleoides, which is the causal agent of the black rot disease in sugar beet. Sources of resistance to this pathogen are scarce. Thus, one of the main tasks in sugar beet breeding is the identification and characterization of candidate genes and QTLs associated with the resistance to this pathogen. In order to achieve this goal, we will perform a transcriptome analysis through next-generation sequencing technique, comparing susceptible and resistant breeding lines. Furthermore, we will assess the population structure of different Aphanomyces cochleoides populations coming from major sugar beet producing areas such as US, Europe, and Japan. The pathogen-host interaction will be also monitored by confocal microscopy to understand the development of the infection pattern.
The Forester Hill campus in Aberdeen, Scotland, is one of the largest medical campuses in Europe, and it includes the Institute of Medical Sciences. The IMS also includes many specialized facilities such as the Aberdeen Proteomics Facility, the Mass Spectrometry and Analytical Facility, the Microscopy and Histology Facility, the Aberdeen Biomedical Imaging Centre, and the Scottish Biologics, which creates custom antibodies. The Aberdeen Oomycete Laboratory is based on the IMS, and it is directed by Professor Peter van West, the director of the International Centre for Aquaculture Research and Development and Chair in Mycology at the University of Aberdeen. His group includes specialists in taxonomy, ecology, epidemiology, bioinformatics, biochemistry, immunology, cellular and molecular biology, and oomycete host interactions. In this laboratory, you will find the tools for a wide range of techniques, including culture of a wide variety of microorganisms and cell lines, recombinant protein production, genome sequencing, gene silencing by RNAi, and in vivo and in vitro host pathogen interaction models. Magdalena Silak is the PhD student of the PROTECTA consortia developing her project in Peter's group. Wageningen University and Research is a National Agriculture College. The Plant Breeding Department is a collaboration between the business unit Plant Breeding of Wageningen Plant Research and the chair group Plant Breeding. The research is organized in 5 main groups with emphasis on crop plants, Brassicaceae, barley, potato, tomato, lily, and rose. The main groups are growth, development and quality, bio-based economy, biodiversity and genetic variation, abiotic and biotic stress resistance, and quantitative aspects. Dr. Vivian Vleeshaubers is leading a group on effector-assisted breeding. Her research aim is understanding the molecular interaction between potato and its pathogens, in particular the oomycete Phytophthora infestans and the early blight fungus Alternaria solani, with the ultimate goal to achieve a more durable and broad-spectrum resistance in potato. Pioneered with effectoromics approach for unraveling new functions of Phytophthora infestans secreted proteins. Her research was expanded to apoplastic immunity and identify ELR, the first potato pathogen recognition receptor against Phytophthora. The laboratory has labs for biochemistry, tissue culture, cytogenetics, botany, and molecular genetics. Main techniques used in the lab are developing molecular markers, selection, and transformation. Further experiments can be conducted in growing cabinets, climatic rooms, glass houses, plastic tunnels, and gauze compartments.
Hello, my name is Tatiana and I am from Valencia. I studied a Master in Research and Development in Biotechnology at the University of Valencia. After my master's studies, I did an internship at a plant biotech company in Wageningen, which introduced me to my actual position as a PhD candidate at the Wageningen University and Research. My project is more fundamental based, and its aim is to understand the interaction between potato and Phytophthora infestans at the intercellular space. In this context, I would like to study the pattern recognition receptors that recognize apoplastic effectors from this pathogen. The identification of these cell surface receptors will contribute to breeding for late blight-resistant cultivars. To achieve this, I will use a genetic approach to identify a new apoplastic receptor, and I would also perform biochemical studies such as co-IPs followed by mass spectrometry to identify its interactors and also FRET to characterize these interactions. Afterwards, this data will be exploited to identify new mechanisms of host resistance breeding to reduce dependence on chemical pesticides.
The research laboratory in vegetal science is located in the southern France near Toulouse. It is part of INRA-E campus, which is the largest agricultural research center in Europe. Since its creation, the laboratory has diversified its research topics in the field of plant biology and plant microorganism interactions. The laboratory consists of 8 research teams, with the main focus on research in the fields of biology, plant microbiology, and molecular ecology. The group studying plant microbial interaction hosts 2 PROTECTA PhD students. The group uses model plant Medicago truncatula to study genetic bases of quantitative resistance, connections between immunity and symbiosis, and mechanisms of plant-host interaction and communication of microbes in soil. The two PhD students in the consortia are supervised by Bernard Dumas and Elodie Gaulin. Their research is focused on molecular characterization of plant microbial interaction using the model plant Medicago truncatula endangered pests Aphanomyces. In addition, this lab has a shared facility with Biocontrol Innovation Center of French private company Desangos. The techniques used in the lab comprise microscopy, oomycete culture collection, metabolomics, sequencing, bioinformatics, field, and greenhouse trials. Tuscia University is located in the town of Viterbo, around 100 kilometers away from Rome. The Department for Innovation, Agri-Food and Forestry is divided into 4 scientific areas, which are complementary and mutually reinforcing. It is a research and multidisciplinary laboratory focused in exploitation, conservation and management of biological systems and forestry resources, food processing and security, human health and chemical processes for the environment, with special attention to environmental sustainability. The Plant Pathology Laboratory is equipped to study the relationship between plants and microorganisms. The disease diagnostic testing is done via isolation of the pathogen on artificial substrates, identification and characterization by molecular assays. Professor Ana Maria Vetraino, an expert in the biology and management of plant pathogens, main supervisor of the PhD student Dania Tabet, is participating in national and international research projects, with many publications related to monitoring and evaluation of Phytophthora community in natural environments.
My name is Dania Tabet from Lebanon, holding a master's degree in integrated pest management from the Mediterranean Agronomic Institute of Bari. My research project is part of World Package 8, Ecology, Diversity and Management of oomycete disease, with focus on the use of plant extract and microbial agent as eco-friendly control strategy against Phytophthora species in nursery. It is a sustainable alternative for synthetic fungicide in Phytophthora control to reduce the hazardous impact of chemical fungicide in human health and environment. For developing an integrated eco-friendly control management method against Phytophthora species, different disease control methods will be integrated. Plant secondary metabolite extracted from dietary and medicinal plant source and microbial agent will be tested under different climate conditions, with the main objective of promoting the awareness of grower, researchers, students and policymakers on sustainable use of natural pesticides.
Oxford University is also part of the consortia. In the plant science department, different disciplines from plant sciences converge, such as biochemistry, cell and developmental biology, ecology, and evolution of plants. Within it, we can find Professor Renier van den Hoorn's lab. This group focused in the biochemistry behind the activity of specific immune plant proteins. Those proteins can have a protective function against plant pathogens. In his lab, the PhD student Laura Ossorio Carballo is investigating the molecular mechanisms underpinning manipulation of those proteins by the plant pathogenic oomycete Phytophthora infestans. The lab has a unique approach to display those protein activities that, combined with powerful functional proteomics technologies, offers an exciting platform for collaborations. Hello, my name is Laura Ossorio Carballo. I'm coming from Spain. I'm studying my PhD at the University of Oxford, and my thesis is mainly focused in Phytophthora infestans, which is the organism causing the potato blight disease. And it is a very interesting organism because it has a complex life cycle with a biotrophic and a necrotrophic phase, meaning that it can live either in a live tissue or in dead tissue. And it also has a fast evolving genome that allows it to overcome any resistance that is being introduced in the cultivars quite quickly. Founded in 2007, Solinta is a true potato seed breeding and biotechnology company based in Wageningen, the hometown of one of the leading agricultural research institutions in the world. Solinta has made diploid hybrid breeding possible in potato, In traditional tetraploid potato breeding, each breeding cycle starts with a cross between heterozygous tetraploid varieties to generate hundreds of thousands genetically diverse seedlings, from which a new cultivar is selected. Disadvantages of this approach are the low genetic gain in each lengthy breeding cycle, the slow reproduction of seeds tubers per season, and the build-up of diseases in the tubers during clonal propagations. Solinta has successfully introduced a self-compatibility restorer gene into potato to enable hybrid potato breeding. Hybrid cultivars typically produce higher yield and show high crop uniformity. Hybrid breeding systems are fast and efficient, and new traits can rapidly be introduced by marker-assisted integration. Two additional advantages for potato are much faster multiplication of hybrid seeds and simpler logistics, since true seeds are easier to ship, easier to store, pristine and not perishable in comparison to seed tubers.
Hi, my name is Max Ossenbrink. I studied horticultural sciences at Leibniz University Hannover in Germany. After graduation, I've been working in a software company in Cologne that develops software for agricultural field trials. Field trials is also what I'm involved with here at Solinta. Solinta is a company that made hybrid breeding in diploid potato possible. And one of their goals is to introduce durable resistance to Phytophthora infestans into potato. Phytophthora infestans causes late blight, which is the most important potato pathogen, and it is unfortunately notorious for breaking resistance genes very quickly. And through crossing with wild species, several resistance genes are stacked into commercial or pre-commercial potato cultivars. And my project is to validate the effectiveness of these stacks or these stacked gene combinations in field trials. Additionally, I'm also involved with mapping studies to find markers for these resistant genes to assist in hybrid breeding. Thank you very much.